Hello, this is Jack Hoppuff, and I'll be describing translation. Translation occurs in the ribosome. mRNA, as we talked before, is the code for a specific protein. mRNA is grouped from 3 bases, which is carried by codons. It just carries codons. These will code for specific anticodons, which are carried by the tRNA. Each tRNA is covalently linked to a specific amino acid. It's arranged of nucleotides in the codons, and it's arranged, and it's called the reading frame. There's a total of 64 possible codons, as you can see. Multiple codons will result in the same amino acid. Each codon codes for a specific amino acid, such as like CCU, CCC, CCA, CCG will all result in proline as your amino acid, but they're all specific in the mRNA processing. AUG, there's like specific special ones, such as AUG. It gets you methionine, which is your start codon. And then as you can see, there's UAA, UAG, and UGA, which is your stop codons. We're gonna just kind of draw it out here. So this is our ribosome, and this is our small ribosomal subunit, which will be on the small part. Small ribosomal subunit, UNIT. And this is where our codon, our mRNA, is going to be attached. So for example, our start will always be AG. And then I guess we can do CCU, and then we'll do like UCU, UAU, and UGA, sure. So this is our mRNA, which is read from 5' to 3'. And once the tRNA enters, it's going to enter through the large ribosomal subunit. Oh, wow. So the tRNA that corresponds to the next codon enters the ribosome. So AUG, it's going to have a tRNA that corresponds and will have an anticodon of UAC, which is the anticodon which helps it connect there through a covalent bond there. And it's going to give us methionine, MET for short. And so as this moves along, I guess we should add that this will be our P group, A group, E group. As it slides down, the next amino acid will enter and connect itself to the methionine. This anticodon tRNA will actually exit the ribosome, leaving just the methionine. So CCU corresponds to proline, which is just PRO for short, and we have an tRNA anticodon of GGA, and it just keeps that process going, and it just slides down, enters in the A. The P is where the amino acid links to the polypeptide chain, E, and it just exits out the ribosome. And as we keep going down, you get, I guess we'll just do them all here. UCU, you get AGA. This anticodon would be AUA. This anticodon, which actually we don't need because this is special for our stop codon. So once it reaches this, proline, UCU corresponds to cernine, which is SER for short. UAU corresponds to tyrosine, which is TYR for short, and that is our new polypeptide chain. Once this is finished, once it gets to the stop codon, this polypeptide chain now will exit the ribosome, and it'll go to an organelle somewhere in our cell. The polypeptide chain goes to another organelle, and it's just going to go through like some folding and some modifications, and we have built a protein. This is basically translation. Thank you.